Sorry, I forgot to make it as a slideshow. So can you see the slideshow now?
Yes, Doctor.
All right, good. Okay, so have you designed primers before? Have you designed primers before? No? Any of you have designed your primer for your final year project? No? Okay, so basically primer design is very simple, it's quite straightforward. There are many available online site that you can go and then have a look. So basically all these programs can be used and available online, okay? So you can go to this link to have a look. Okay, so general guideline for primer design, efficacy and sensitivity of PCR depends largely on efficiency of primers. So the reason why we design primers, because we want to do the amplification, all right? So amplification can be dependent on what is your target. If you are targeting one gene, so you need to design based on the gene sequence. Ability of an oligonucleotide to serve as a primer depends on, the first one is association and dissociation kinetics of primer template duplexes. So this is basically, you have to look at the similarity and dissimilarity between your primer and also the templates. So the template is basically the gene sequence that you are targeting, okay? So this is look at the annealing and excitation temperatures. The second one, duplex stability of mismatched nucleotides and their location. Polymerase efficiency at recognizing and extending a mismatched duplex. So we will go into detail on this. Effective primers, primer pairs should fulfill certain criteria. So including this criteria include primer length, relative percent of GC content, so GC, the G and C bases, melting temperature and annealing temperature, 5' end and 3' end stability and specificity. So the first one, we go into the primer length. So basically the primer length also affects specificity and stability of primer annealing to the template. Okay, so typically you can design between 16 to 30 nucleotide, but normally so far you can also design up to 25, perhaps between 80 to 25. It depends on the properties of the primer itself. Because if you design too long, probably the properties may not be suitable with the appropriate or optimum of the primer's property. Too long increases melting temperature and secondary structure formation, such as hairpin. Too short decreases specificity. Because imagine that if you have too short primer, this short sequence may anneal to any location. So for example, if you have too short, because the possibility of the shorter sequence to be annealed to multiple region is higher. Do you get it? If you have a longer sequence, and then the specificity will be higher because you may be able to be targeting only one specific position. Increases possibility of primer dimer and non-specific binding. So if you look at this, you can see that based on these properties, so normally primer design program will come out with the properties of the primers that the program has designed. So if you look at here, the melting temperature, so you can see this is 42 degree, 62 degree, and then the salt concentration, the primer, the percent of GC, and the length. So this is all been given by the program. Okay, so you can refer to this property whether it is suitable for you or not. Avoid long runs of a single nucleotide, so as this may increase slippage problems. So a long run of single nucleotide, such as a repetitive of a single base. For example, here AAA, CCC, GGG. So try to avoid this. Unless it's very difficult, perhaps you may have consider perhaps maybe 3 repeats, or like 3 A's or 3 G, something like that. But try to avoid. Relative percent of GC content, an effective primer should contain a percent GC content. So this is the optimum or recommended range. So between 45 until 60%. But sometimes the sequence might be very difficult to get the primer sequences that have within this range. Perhaps it's too much of GC content in that particular sequence. So you may consider to extend a bit the percentage of the GC content. Okay, am I talking too fast, Faizatul? Just fine? Because you look confusing. Just let me know if I'm talking too fast. Okay. GC content provide information regarding strength of annealing. Okay, as you know that GC bonding is 3 hydrogen bonding as compared to A and T. So that's why GC content reflect the strength of the annealing. Okay, if a primer has low percent of GC content, it extend the length of the primer to adjust the melting temperature. Okay, so that's why try to design according to perhaps 18 to 25 nucleotides, and then if the GC content is too low, then you can extend and make the primer sequence a little bit longer, perhaps until 26 or 27, okay, to increase the GC content. GC should distribute evenly throughout primers, and then you have to avoid long GC runs, okay, especially at the 3' ends. Melting temperature and annealing temperature. So basically melting temperature is when the DNA strand become single-stranded, okay, but only at the half of the region, okay. And then another half of the primer sequence will be still double-stranded, okay. If you look at this graph, you can see that the melting temperature usually indicated by the 50% of the single-stranded cDNA nucleotide. Okay, so this is where the melting temperature is identified. Annealing temperature, this is when the DNA strand anneal to a template. So DNA strand meaning that the primer, melting temperature is usually higher than annealing temperature. So this is basically a simple equation how to calculate the melting temperature. So basically, how many A and T in the primer sequence times 2 plus how many G and T times 4. Okay, so it's quite direct. And how you can calculate the annealing temperature is by subtract or minus melting temperature minus 5 degrees. These temperatures for each primer should not exceed optimal polymerization temperature of the polymerase. So usually the polymerase do the polymerization at 72 degrees. So make sure the melting temperature or the annealing temperature are not more than or higher than 72 degrees. So optimal melting temperature 52 until 58. So primers with lower melting temperature has lower efficiency. This may produce non-specific PCR products. So you probably want to increase your melting temperature. Okay, so normally once you design a primer, you will do gradient PCR. So gradient PCR meaning that you set the PCR reaction at different temperature. So in this case, perhaps you can set 52 until 60, for example, and then you will get different PCR product. One PCR product that amplify with melting temperature at 52 degree, the second one probably at 53 degrees, and so on. So at each different melting temperature, you will get different band. Okay, the band could be only one or could be more than one. If you get more than one, meaning that you get non-specific binding or non-specific PCR products. So you don't want that, so choose the reaction with melting temperature with only one PCR product or only one band. Okay. Melting temperature higher than 60 should be avoided. So this may increase secondary annealing. So normally it depends on the reaction that you want. You may go up to 60 because sometimes the primer is efficient at 60 degree. So you may opt to go for 60 degree. Okay. 5' end and 3' end stability and specificity. So the 3' terminal position is essential to control the mispriming. Okay, so primer should be stickier at the 5' ends than the 3' end. So a sticky 3' end is indicated by a high GC content. As I mentioned earlier, you should avoid too high GC content at the 5' end as compared to the at the 3' end as compared to the 5' end. Okay, so the first one that you have to look at is the GC clamp. So this is basically based on the presence of G or C within the last 4 bases from the 3' end of the primer. Sometimes you may get information saying that the last 5 bases. So it should be similar, but as long as you look at the 3' end. Okay, essential for preventing mispriming and enhance specific primer template binding. So looking at this, you have to avoid more than 3G or more than 3C or more than 3 plus C. Sorry, more than 3 of G and C. Okay, at the 3' end. Maximum 3' end stability. So stability of the 3' ends look at the delta G or the Gibbs free energy. Okay, so delta G depends on the Gibbs free energy that measure the amount of works that can be extracted from a process operating at a constant pressure. So it looks at how basically it can release the energy in order to dissociate between these primers and also the template. While higher prime end stability improve priming efficiency. Okay, too higher stability could negatively affect specificity. Okay, so this is basically look at the hybridization. Okay, and then it can cause the non-specific extension. All right, so in this case, avoid delta G with the value less than -9. Okay, so the if you look at here, avoid delta G less than -9, meaning that consider as -8, -7, and so on. All right. Oops. Hairpins form via intramolecular interaction. So these are examples of how hairpins structure could look like. Okay, it can affect the primer template binding. So this can cause poor or no amplification. Okay, so this is basically acceptable delta G when you have, so basically as I mentioned, delta G is free energy required to break the structure. So the acceptable delta G is more than -2 for 3' end hairpin or more than -3 for internal hairpin. Okay, so this is basically considered as internal hairpin, and this is 3' hairpin, 3' end hairpin. Okay. Self-dimer, so this is when you have the primer anneal to the primer itself. Okay. Intermolecular interaction between the two same primers. Acceptable delta G more than -5 for 3' end self-dimer and more than -6 for internal self-dimer. So this is the 3' end self-dimer and this is internal self-dimer. Okay.
Doktor,
Yes.
apa beza internal hairpin dengan lagi satu tu?
Internal hairpin. So this is basically look at the position of the hairpin. If the position of the hairpin occur at the end of the 3', so meaning that is considered as the 3' end hairpin. Okay, so if you look at here, this is internal because it is at the end, but it's not exactly at the last nucleotide. Sometimes the hairpin may occur at perhaps at the 3 position of the nucleotide sequence. So perhaps it start from AAA, for example. Okay, so that considered as internal. So this is, as you can see here, at the end of the nucleotide sequence. Okay, the first nucleotide from the 3' orientation. Okay. Okay, cross dimer, so this is called as heterodimer. So this is when the dimer are between different forward and reverse primers. Okay, so as you can see here, forward and reverse. So acceptable delta G more than -5 for 3' end cross dimer and -6 for internal cross dimer. Okay, similarly, so you can see this is internal or end cross dimer. This is internal or end cross dimer? I'm asking you.
Internal?
Yes, this is internal. Okay. However, sometimes this secondary structure are harmless because the annealing between these primers, they are occur below the lowest temperature that have been set in the PCR reaction. Okay. For example, some dimers or hairpin formed at 30 degree, but during the PCR, the lowest temperature set at 60 degree. So probably you will still be getting the PCR products. Okay. But still you need to avoid. Other consideration, primer pair matching. So basically, in principle, primers work in pair, but sometimes you can run a PCR using only one primer. Okay, you can choose whether you want to run using the forward primer only or reverse primer only. But it may produce only the one strand, not the both sense and antisense. Okay. Since they are used in the same PCR reaction, it shall be ensured that the PCR condition is suitable for both of them. So when you want to design the reverse and forward primer, make sure that the maximum difference of the melting temperature is up to 3 degrees. Okay, the closer the better. All right. Summary. So basically, when is a primer? A primer. So if you look at this figures, you can see that if everything is fine, similarity between the primer that anneal to the template, then it will amplify. What about here? Only the region from 3' until perhaps half of the primer is still will do the amplification. And this one as well, it can also amplify the sequence. That's why it's very important to design the primer very specific to your template. But if your 3' end doesn't anneal to your temperature, to your template, and then the amplification may not be occurred. Okay. So that's why it's very important to look at the 3' end of the primers. Okay, now we look into another type of primers. We call it as degenerate primer. Okay, so how to design degenerate primers? So the first one, you can either this is the first one I mentioned here, as I stated here, as if necessary, because it depends on the objective of your amplification. Okay, if in the case that you only have the protein sequence, you don't have the nucleotide sequence, because in PCR you need to have the nucleotide sequence, am I right? Because you can only amplify the nucleotide sequence, either genomic DNA or cDNA. So if you have the if you have the protein sequence, you can design directly. But if you don't have any information in regards with the target gene or the target protein, so you have to align protein sequences from different organisms that are available in the database. Okay, and then the second one, target a region for amplification. So this is quite similar to what you have done for the previous one when you want to design normal primers or common primers, and position forward and reverse primers in more conserved regions. So this is to ensure that you design based on the information that you have. Okay, and design the degenerate primers. So this is based using specific programs for degenerate primers. Okay. Okay, so basically, degenerate primers are used when only the protein sequence of a gene is known, okay? Or when you want to isolate similar genes from a variety of species. Okay, so what you have to do is basically, as compared to common primer, you have to select 6 or 7 residue peptide sequence. So you have to depend on the amino acid sequences first. Okay, so since one amino acid is similar to 3 nucleotide, right? So that's why by selecting 6 to 7 amino acid residues equals about 20 nucleotide primers. Okay, a degenerate primer is an accumular mixture of a number of primers. This is because of the degeneracy of the codon. So that's why it called as degenerate primers. A 4-fold degenerate primer is made of 4 different primers or oligonucleotides. Okay, so now we look at here. For example, you want to design a degenerate primer at this particular region with the sequence of YEILLK amino acid. Okay, and then the codon for this each amino acid residue, why it can be degenerate from TAT or TAC codon, E from GAA or GAG codon, I, L, K, L, and K. Okay, similarly, all of these amino acid have different codon. Okay, it may be 1 or 2 codons. Usually, one amino acid equals to or encoded by multiple codons. Okay. Okay. Now, the primer sequence or the degenerate primer sequence for YEILL will be TATOC. Look at here. The first 2 nucleotide TA, but the third position is TOC. Okay, so that's why it will be TATOC. And then E, look at the second, the first and the second, GA, but the third can be either A or G. So that's why for E, it will be A or G. Okay, do you get it? And then look at I. The first position of the first nucleotide of I codon AT, AT, AT. But the third one, TCOA. So that's why you can see here the first two are AT, and then the third one could be CTOA. Okay, similarly like LLK here. So that's why you get these different primers sequences at the end. Okay, do you get it? So it could be TAT, GAA, ATC, blah blah, or TAC, GAG, ATT, CT, blah blah. Okay, so that will be multiple primers. So the fold degeneracy will be 2 times 2 times 3 times 4 times 4 times 2. So this is because there are 2 options here, 2 options here, 3 options here, 4 options here, 4 options here, and 2 options here. So the options will be the fold degeneracy multiplied together. Okay, so in this case, you will get 384 fold degeneracy. Okay, so this is a very high order of degeneracy. Okay, is it advisable? No. Okay, so try to decrease the degeneracy by selecting probably different region or choose the region with amino acid with lower degeneracy value. There are codon research tables, so you can find through this website. So this is the website show the frequency of the codon that used in a specific species. So for example here, in red, almost half of codon used for leucine is CTG, while only 7% is CTA. Do you understand? So for amino acid leucine, in red species, the leucine derived from the codon CTG, 50% of them. Okay, another 50% derived from different codon. Okay, this is only specific for leucine amino acid, right? So another 70%, they come from CTA codon. Do you get it? Okay, one can therefore use only 2 or 3 of the 4 possible codon for leucine while designing the degenerate primer. Okay, so in this case, you can also refer to the database. If let's say you have the information on that particular species in the database, refer to that database to help you to design the efficient primer sequence. Okay. The other thing to do will be to align the nucleotide sequence of human, mouse, and horse. For example, back to the peptide sequence YEILLK region. So in human, there are TA, TGAA, blah, 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 in mouse, horse, and rat. So now, if you look at the first 2 nucleotides of the codon, there are TA, GA, similar to the rest. They are the same, okay? And then look at for K. So in here, what we can identify that the codon that code for K is only AAA. So definitely this will be AA. We can assume that rat also has AAA codon. Okay. So in this case, we only need to design the degenerate primer at this particular position and assume this only AAA. All right. The alignment shows the right sequence probably with such, okay, this is what I have explained before. Because if you look at the previous slide, K in this case, okay, K can be AA or AAG. So since the other organism, this K encoded by only AAA codon. So we can ignore this AAAG. So meaning that at this particular position, you can only design as AAA instead of AA, AOG. All right, do you get it? Substitute the inosine for the 4-base wobble. So this is use CTI for leucine rather than CTC, CTA, CTG, CTT. So leucine basically refers to the 4 bases. Okay. All right. This to reduce the number of oligos that have to be synthesized. The consideration in designing the degenerate primer, if you want to amplify similar protein sequences, then you have to look at the most conserved region of the proteins. Okay, if some of the residues are not completely conserved, then the primer sequence will need to accommodate all possible codons of all amino acid residues at that site. Okay, so you can also refer to this table for the designing. So this is degeneracy code. You can look at here the amino acid residues and the symbol that you can use when you want to design the primer. Nucleotide sequence and complement. This is for reverse sequence. Okay. If you see here, this is usually, not usually, this is basically generated based on this. Okay, so assuming that UUU is, this is in the mRNA, but these degeneracy codes are generated from this. Okay, so which codons that can encode serine, for example, for proline. So that's why they come up with the degeneracy code in the previous table. Okay, so if you look at here, the peptide sequence should avoid amino acid that have a lot of codons, such as leucine. So here, leucine derived from 4 codons. And then arginine. So you want to avoid this as well. Arginine comes from 4 plus 2, 6 codons. And then serine, 1, 2, and also another 4. So please avoid this. Okay, and try to get the region with 1 or 2 possible codon, such as methionine, W, C, D, E, F, and so on. Okay, for example here, phenylalanine, only 2 codons. You can target at that particular site. Okay, so this is, for example, if you have, if you don't have any information on the genome sequence of the organism that you want to look at. So first of all, you can do normal multiple sequence alignment. So you will get the MSA like this, okay? And then this MSA will show you different blocks with the conserved region or non-conserved region. Okay, so now you have to target a region for amplification. Okay, so you have to design the primers. So now the question is, which region that you will choose? So looking at these different boxes with different colors, which region that you will choose to design degenerate primers? If your template is unknown sequence, if you have template with unknown sequence. This is a question. So which region that you will choose? Either the green box, the yellow, pink, blue, or purple. Come on. Izani? Izani, are you there? Firdaus, Izani? Are you taking pictures?
I think I will choose the green box.
The green box. Okay, why? Can you turn on your
Because,
camera when answering the question?
because it have more conserved region, as we can see the red sequence.
Yes, correct. In this case, we will go for example, you can choose this particular position or region because they have very high conservation at this particular position. Okay, so this basically can reflect or can show you the domain of these proteins. Okay, because they have high similarity at this particular position. Okay, what about let's say you design forward primer here. What about the reverse primer? Anyone can answer? Nadira Zaidi, are you there?
Yes, Doctor.
Okay, can you turn on your video while answering? Nadira?
I think for the reverse primer, we choose the same sequence.
Sorry, I couldn't hear you. I heard motor, bunyi motor kat belakang. What was your answer?
I think. Can you hear me, Doctor?
For which position, which box that you will choose to design the reverse primer. So now Izani have point out that we can design the forward primer in a green box. What about the reverse primer?
I think the same box,
The what, sorry?
the green box.
The green box?
Yes.
So Izani designed forward primer in the green box. So you want to design reverse primer in the same box, in the same region?
Or maybe the yellow box.
Yellow box. Are you sure? Or you want to go to the pink box? So you stick with your answer, the yellow box?
Yes.
Yes? Okay.
Yes.
Okay, thank you, Nadira. Can you design, okay, I asked someone else. What about Jiashin? Jiashin, are you there? Can you design reverse primer in pink's box? Okay, Izani have decided to design the forward primer in green box. And Nadira suggested to design reverse primer in yellow box. So can we design the reverse primer in pink box?
Yes.
Yes? Okay, are you sure?
Yes.
All right. Okay, thank you. Yes?
Yes, I think can.
Can. Okay, thank you, Jiashin. Okay, so basically, it will be more advisable for you to design the reverse primer in the yellow box. Okay, because you can see that the conservation of this particular region is higher as compared to the pink box. All right. You can design your reverse primer here, but you may be find it is a bit difficult because you can see here high similarity at these positions, but you can see low conservation at this particular position. Okay, so that's why it's a bit difficult here. You may get your degenerate primer with high degeneracy value or high fault degeneracy value. So you may want to avoid this. Okay, but still you can design the primer in this box. Okay, what about Darminram? Darminram? Are you there or not?
Yes, doctor. I'm there.
Okay, can you turn on your video?
Yeah, it's on.
Okay, it's on. I cannot see you. Okay, now I can see you. Can we design the primer in, if let's say we have the forward primer in green box, can we
Okay.
design the reverse primer in purple box?
To my personal thing, I think it's not really advisable.
It's not what? Sorry, come again?
Not really advisable.
Okay, why?
I feel there's less rate conservative sequences there.
Okay, yes, that is correct because it's too, the conservation is too low here. Yeah, all right. That is correct answer. And there is another reason why you may want to avoid to design reverse primer at this region. Can someone else give me the answer? Thank you, Darmin.
Thanks, doctor.
Sabrina, MK, are you there?
Yes, doctor.
Okay. So basically, Darmin said that it is not advisable to design the reverse primer in this region because the conservation is very low. Other than that, why it is not advisable to design the primer here? Another reason.
I'm not really sure because my reason would be the same because the conserved region is less compared to the other boxes.
A similar answer. You give me the similar answer. Okay, what about the length of the primer product that will be generated?
It will be too long. It's like at the green and the purple.
Yes, this will be too long between this particular region until here. Okay, you may want to consider looking at suitable length of the PCR product that you want to target. Okay, thank you, Sabrina. But I'm not saying that you cannot design the primer from at the end here until end. You can do so, okay? But make sure that it is possible and using a suitable polymerase, okay? All right, good, very good. Okay, so primer design example. So this is basically, we want to look at how you can design a primer for T1 lipase from Geobacillus zalihae. So I think you have been working with this sequence for your practicals, if you remember. So this is lipase protein that isolated from these microbes. And this microbe was isolated by Prof. Zaliha. So that's why it names as Geobacillus zalihae. So this is work done by Dr. Adam, if I'm not mistaken. Okay, so now, for example, we have the sequence. All right, so in this case, we have the cDNA sequence. As I mentioned earlier, you can do the PCR on cDNA or genomic DNA. So the primer need to be designed depending on the template. So if the template is cDNA, then you have to refer to the transcript sequence. But if your template is genomic DNA, you have to design the primer based on genomic DNA sequence. Okay. How do we design 22 nucleotide primer pair with so this one we want to insert the RE site, restriction enzyme site. Okay, so determine hybridization site, design primer sequence for hybridization, insert RE sequence, evaluate parameters of primers or the properties of the primers. So now we start to by choosing the position. So we want, in this case, we want to design the primer and we want to amplify the template from end to end. So that's why we target from here until the end. Okay, so can you design the primer from 301 until here, for example? Can you do so, Maizatun? Can you design from this, let's say you design forward here and you design primer here, for example. Can you do so, Maizatun? If you want to design the whole sequence. Do you get my question?
Yeah, I get it. I think not primer at the end because there will probably you won't get the upstream of the sequence.
Yes, correct. The sequence will not be full length. Okay, because you only aim the internal sequence. So, okay, identify forward and start position, and then forward primer sequence is usually 5' to 3' of the first few sequences of the gene. So in this case, we aim at this particular region. The reverse will be the last few sequences of the gene with reverse complemented. Okay, so if you look at here, this is the reverse sequence. But the primer will not be GTCC, GCCA, and so on. This reverse, this region need to be reverse complemented. Okay. So, okay. So design primer for hybridization, the forward is quite straightforward. So your the forward sequence will be 5' to 3'. So in this case, which one will be your forward sequence? The black one or the red one? Subatra, Naya, so which one will be the forward sequence? The black one or the red one? What are you looking for? So which one will be the forward sequence?
Doctor, she posted the answer in the comment.
Okay. Izani is the name of his father, doctor. Oh, sorry, Izani. Black one, doctor. Okay, yes, yes. That is correct. Sorry, ya Izani. Sorry, Izani, ya Allah. So, his name is Firdaus. Yeah. Okay, why you didn't mention it earlier? Firdaus ni pun. Okay, alright. So, the reverse sequence. So, which one will be the reverse sequence? The black one or the red one? Sharifah Fatin?
Red one, doctor.
Yes, because it's the reverse complemented sequence. Okay, so it's quite simple. So this is an example for the PCR primer program that you can use. In here, we shared on Primer Blast. This is very, it's quite easy to be used. So you just need to key in the sequence and then you can decide the range from the position of the template sequence. Okay, so you can decide whether you want to start from 1 to 50, for example, and the reverse is located at, in our sequence, in here, you can decide 1921 until blah, blah, blah. Okay, so make sure that the range within the sequence length. Okay, so this is when, if you have your own forward primer and reverse primer sequences, you can also put here to identify the position of your primer and also the properties. And this is all the parameters. So please go to this website and you can see that there is also parameters that to check the specificity of the primers. Okay, when you want to do the amplification of the gene, your template comes from a collection of genes or collection of transcripts. Okay, because in genomic DNA, there are many genes, as I showed you before. They could be thousands. So you need to check your primer whether there are specific target to that specific gene that you aim for. Okay, so similarly to the mRNA. So you can check and choose the database related to your template. You have learned on the databases, right? So the transcript, the mRNA and the the gene RACSec, for example. So you can choose from that. This is also called primer validation. You can also blast the primer sequence against a whole genome sequence to ascertain the uniqueness of your primer. So this is what I was talking about. So this is the result. Basically, you can see the properties of the primer with the length, the start and stop, the position of the primers, the melting temperature, GC content, self-complementarity, self-3' complementarity, and so on. Okay, it could be more than one pair primer sequences that the program will generate. Suitable calculated melting, so this is you have to check whether it is okay or not, and then sometimes you have to use other program to define whether your primer may not be able to form secondary structure. For example, hairpin or loop hairpin. Okay, you can learn it during the practical later. Suitable calculated melting temperature. So this you have to look at whether it is okay or not. And then insert restriction enzyme, betul lah, sequence site. Okay, so in this case, we have decided what is our forward and reverse sequences. So now we want to insert our site for XH01. So this is the sequence. So we just need to add in. Okay, for example here and here. All right. Okay. So this RE site, when you add in RE sequence in your primer, your PCR product will end up with having the restriction site at both ends. Okay. If you only put one site with different enzyme sequence, then it this will be different lah, from one end to another end. Okay, do you get it? Yes or no? Yes or no?
Yes, doctor.
If you don't understand, you can just ask me. Okay, check parameters. So this is based on what I have explained before. If too high and then decrease the primer length. So if it's too low, increase the primer length. So I have a question. Another question. I'm looking for a victim. Aisyah Adila. Aisyah Adila, are you there?
Yes, Doctor.
Okay, can you turn on your video, please? Looking for shawl first.
Yes, Doctor.
Okay, berjaya dapat tudung Okay, so my question is, can let's say you use a program to design the primer. Okay, so now looking at the temperature. Okay, and then if it is too high, you can decrease the primer length. If it is too low, you can increase the primer length. Can you extend the primer sequence although you have used PCR primer program to design the primer? Do you get my question?
Can you repeat?
Okay. For example, in this case, you get the primer sequences for the first one and the second one. Okay. If you look at the melting temperature, it is too high. Am I right? Is it too high or not? Okay.
Yes, Doctor.
Okay. So based on this, do you need to redo the primer design, or you can just cut from this primer sequence?
I think just cut the time sequence.
Yes, you can also just cut the primer sequence, but sometimes it may reflect the percentage of GC content. If you cut here, for example, you cut from here until here, so the melting temperature will be lower, but your GC content will be lower as well. Okay, so what you can do, if the percentage of GC content is too low, can you move to other position at this end? Although you cut here, you cut here to reduce the melting temperature. But now what happened? The percentage of GC content also become very low. For example, less than 45%. Okay. So can you extend the primer at this upstream region?
I think cannot.
Cannot. You think cannot? Actually, you can. Okay. You can go back to the template sequence and then extend a little bit longer. Perhaps there are containing different nucleotides, okay, that reduce the melting temperature and also increase a bit the percentage of GC. Okay. All right. Thank you, Aisha. I have question to Muhammad Farkan ke? Is it your name? Muhammad, I can see your name Muhammad Fak je. Sorry, Muhammad Farkan Oh, Muhammad Fakrul. With the glasses. Okay, my question. My question is, I haven't asked yet. My question is, can you design your primers manually by looking at the sequence, the template sequence? So for example, you don't have access to the programs. So can you design the sequence based on this template sequence only?
Yes, I think so. We can design it manually.
Okay, yes, you can design it manually. but still you need to validate the primer that you design, whether it is okay or not. All right. Thank you, Muhammad. So your name is Muhammad Farhan. Oh, Fakrul.
Fakrul.
Oh, Fakrul. Okay. Okay, thank you. All right. Okay. So this is basically when you want to design the primer to amplify the whole length. Okay. So in this case, the primer will be from here and here. Okay. If you want to do gene expression analysis, you can design only at certain region. You can focus at the middle of, in the middle of the sequence. Okay, you don't have to design from end to end. Okay, that is the case of gene expression analysis. Okay, and then the last one will be just the tools, the other primer design tools that you can use. Okay, any questions so far?
Doctor, I have a question.
Yes, who is speaking? Okay.
Kai Fong.
Okay.
Just now, Doctor has said our PCR product cannot be too long. So what is the optimum length of the PCR product?
Optimum length of PCR product. So basically, I would say there is no optimum length for normal PCR product. It depends on the tag polymerase that you use. Usually, the common tag polymerase can go up to 5 kb. So the amplicon or PCR product can go up to 5 kilobase per. All right. But if you want to design the primer for gene expression analysis, for example, you want to use real-time PCR. Do you know real-time PCR?
Yes.
Okay. So for real-time PCR, it is advisable to go from 100 until 300 amplicons. 100 until 300 base pair of amplicons length. So it depends on what you want to use and what you are doing. Sometimes, what happened in my case, my master student, she was working on isolation of a gene from a plant without any genome sequence information. So that's why she needs to design the degenerate primer. But she tried to design from one end to end, but she couldn't get it. It's very difficult. So that's why we just look at the conserved region first. Once we get the sequence in the middle, we target for the upstream and downstream region by doing the walking PCR. So we design side by side, region by region. So back to your questions, it depends on the tag polymerase that you want to design. Sometimes the tag polymerase kit, it mentioned that it can go up to 5 kb, but it sometimes it may not be successful. So you probably need to do a lot of optimization, either redesigning the primers or optimize the PCR reaction itself.
Okay, thank you, Doctor.
All right.
Doctor, I was curious, for full genomic sequence, do they actually only use forward and reverse sequence, or do they actually use forward and reverse sequence in different sections so that they can get like different parts of the genome and then they combine it into one?
Actually, you can do so by doing the PCR individually. Run one reaction with forward primer and another one reaction with only the reverse primer. You can do so actually, and then you have to do genomic sequencing. You cannot combining the applicants from one to another one. Do you get it? So you cannot, you can do annealing. So you have like, it's quite complicated to say. Theoretically, perhaps you can do annealing between the forward result with the reverse result. So probably they have overlapping at the end. But practically, to my knowledge, there is no such thing that we are doing. So you can do just by doing on the sequences. You have learned this in previous practicals, right? So you get the forward sequence and then the reverse primer sequencing result. So now you can do the alignment to see the full length. Okay. Originally, in the sequencing analysis, that's what they do. They do the PCR sequencing by using the forward primer and another one by reverse primer. Okay, so separately.
Thank you, Doctor.
Okay. All right, any other questions? Nope. Okay. So okay, so now what you need to do is the practicals as usual. So this is quite easy. I have shared with you the practical. So can you see the manual? Okay. So the topic primer design, so the learning outcome, design forward and reverse primer sequences, evaluate suitable parameters for the design primers. So this is quite straightforward. You just need to go through all this step by step. And then just an additional part where you need to do the validation of the primers that you have designed. So here you are using Primer Blast. Usually Primer Blast will give, you may not know what is the parameter to determine whether the primer sequences can produce hairpin or secondary structure. So it depends on the self-complementarity. Okay, so Primer Blast usually set up with less stringent to determine whether the primer sequences may have secondary structure. So that's why we add in another oligonucleotide property calculator. So from this website, you can see this, right? So you can go to this link and then you need to check whether the primer is okay or not. Okay, so then you will see what is your potential hairpin formation, whether it is occur or not. Okay, so this is quite simple. What you need to do is you don't have to screen step by step what you have done, but what I want you to do is screenshot the output and then give me the answer. Okay, so you look at here. So this is basically the steps. And then once you get the output, you screenshot, put it in your practical report, and then give me the answer. Okay, so this is quite straightforward. You should be able to do it. And then,
Doctor,
yes,
we just need to answer in point form or paragraph?
Point form. Point form. Okay.
Doctor, for
Yes.
this report, is it supposed to be a group work, pair work, or individual?
Individual. The last one, the last practical. Okay, so if you look at number 7, set the range of the forward and reverse primers. So this is, you can set any range that you want. Okay. If you want to target in the middle and then go for it. If you want to target end to end, go for it. Okay. All right, I have a question. Why do you think, what is the application by inserting the RE site in the primer? Anyone want to answer before I pick someone?
Can I try the guess?
Yes.
Maybe it's so that, for example, if you want to target the middle part, so we can just cut off from the primer part, the front part, and then primer, and then we sequence, and then the end, and then cut it from there. I don't know. My guess. That's why
That is incorrect.
Okay.
Doctor, is it
Yes.
We want to insert it into the plasmid so that we can do the cloning?
Yes, correct. We can either do this for cloning to basically, so we need to have this RE site. So it need to be complement with the vector that you want to use. Okay. Other than that, you can also use this for site mutagenesis. So site mutagenesis, when you want to have one nucleotide different in the sequence. So the different will generate different amino acid. So this is one example if you can also apply to your primer sequence. Another application. Okay. Alright, so any questions so far? Ada tak lagi?
Doctor.
Yes.
Our lab report, right? We need to do those introduction, methodology, everything, or we just summarize on the results.
Just give me the screenshot of the output and then the answer.
Okay.
No need introduction and so on.
Okay, thanks, doctor.
Okay.